Hi, in this video I will be discussing some of the questions from sexual reproduction in flowering plants. Sexual reproduction flowering plants. So this video is basically for KCET exam. So read out the first question. Sporopollenin is a chemical substance found in. So sporopollenin, it is a chemical substance. It will be found in, as we all know. So consider this as a pollen grain. So pollen grain has two layers. One is intine, another one is exine. So this intine, it is a continuous layer and it is mainly formed from cellulose and pectin, whereas this exine contains a material, or it is formed from a chemical substance, that is sporopollenin, which is highly resistant to any organic material. It cannot be degenerated by any of the substances like alkaline solution, enzymes, etc. So due to the presence of sporopollenin on the exine, so it will remain as, or it can be remnant as a pollen grain for a long period of time. So this is a condition where pollen grains can remain as a fossil. So here sporopollenin will be seen in the exine of a pollen grain. So read out the next question. The following is the diagram of TS of anther. Identify the parts labeled A, B, C. Here they have given the structure of anther, that is TS of anther, the structure of microsporangium, the TS of anther. Here, all you need to do is you have to label the things given in the diagram A, B, C. Here A stands for this one. It is nothing but the connective. So here TATS of anther, it has four different layers: epidermis, endothelium, middle layer, and the innermost layer, that is a tapetum. B, it will be a endothelium. Then this C, these are nothing but the pollen grains. So right answer for this question will be A, connective, B, endothelium, C, pollen grain. So one is the right answer for this question. Then read out the next question. Match the entries in column 1 with those of column 2 and choose the correct answer. Here they have given the name of the pollination in the column 1. In column 2 they have given type of pollination. So name of the pollination will be a cleistogamy, geitonogamy, entomophily, and xenogamy. Cleistogamy, as it is a closed flower, the flower which will not be open, where a self pollination, that is autogamy, will be observed. So for this one, pollination between the flower, no. Zygomogamy, where the pollination will be seen in the bud condition, that is bud pollination. Zygomogamy, the pollination between the flowers in the same flower. Entomophily, entomo means insect, so that is insect pollination. Xenogamy, it is always cross pollination. So for this one, the right answer will be A, N, B, O, C, M, D, Q. So 3 is the right answer. Read out the next question. Again, in this question, they have given a diagram. All you need to do is labeling the things. A here, which is given here, that is outermost part, that is calyx, that is sepals. Then B, they have given a pistil, the centermost part, stigma, style, ovary, so that is a part of the pistil. Then C, it is a stamen. D, it is a petal. So right answer for this one will be sepal, pistil, stamen, petals. So 3 is the right answer. Then read out the next question. Edible part of the fruit of apple is. So fruit of the apple, it is considered as a false fruit. False fruit, where the fruit is mainly formed from the accessory part, that is the thalamus, other than ovary, that is thalamus. So in case of apple, the thalamus is the edible part. So read out the next question. The embryo sac of an angiosperm is made up of. So typical angiosperm embryo sac, it is 7-celled, 8 nucleated. 7-celled, 8 nucleated. So let me draw a diagram here. So in chelagel end you will come across 3 antipodal cells. These are called as antipodal cells. Then in micropellar end you will come across 2 synergid cells and 1 egg cell. In the center you will come across 2 nuclei. These are nothing but the polar nuclei, and this is a central cell. So this is a central cell. So in here antipodal cell, they are 3 in number. Central cell, it is 1. Polar nuclei, then here you will come across 3 cells. 3, 6, 1, 7 cell. 8 nucleus, 1, 2, 3, 4, 5, 6, 7, 8. So 8 nuclei. So typical angiosperm embryo sac contains 7 cell and 8 nucleated. Then read out the next question. Again they have given a diagram of a monocot embryo. You have to label the parts here. D, it is a scutellum. F, it is the shoot apex. C, it is coleoptile. A, epiblast. B, radical. Then E, coleorhiza. So based on this diagram, you have to choose one option. A, epiblast. B, radical. C, coleoptile. D, scutellum. E, coleorhiza. F, shoot apex. So fourth option is the right here. So read out the next question. So I have picked some of the questions from previous year KCET. In castor and maize plant, they have given this statement clearly in the outbreeding devices. castor and mesh, where you can prevent autogamy but not zytonogamy. So autogamy can be prevented but not zytonogamy. So 1 is the right answer for this question. Next question. With respect to angiosperms, identify the incorrect pair from the following. Incorrect pair from the following. Antipodals. Antipodal cells. Antipodal cell. It is N in number. As embryo sac is N, the cells which are present in embryo sac, that will be N, not 2N. So this one is right here. Vegetative cell of male gametophyte, it is N. It is also true. Primary endosperm nucleus, that is 3N. It is also true. Cells of nucleus of ovule, that is 2N. The nucleus, the ovule contains a number of cells, that is nucleus. It will be 2N in number. So right answer for this question will be 1. Antipodal cell, it is N. The 200-year-old seed excavated from King Herod's palace at Dead Sea belonged to. So 10,000-year-old, they have given 2 examples in your NCRT. One is Lupine articus, another one is Phoenix dactylifera. Lupine articus, it is it is 10,000 years old, whereas Phoenix dactylifera, it is a recently got excavated by the archaeologist in Dead Sea near King Herod's place. So D is the right answer. That is 4 Phoenix dactylifera. It is 200-year-old seed. So seed viability will be varied in different species of the plants. In case of Lupine articus, a seed can be viable for 10,000 years, whereas in case of Phoenix, it will be 2000 years. The next question. Seeds without fertilization is obtained from. Seeds without fertilization is obtained from, or this process is nothing but apomixis. Parthenocarpy, fruits formed without fertilization. Polyembryony, more than one embryo, that is polyembryony. Dormancy, the seed will remain unstable, or seed will not show any activity, that will be considered as dormancy. Here the seeds without fertilization is nothing but apomixis. The next question. The edible part of fruit, already this question has arisen. So that is thalamus of the apple is the edible part in case of the apple. The next question. In a dithecous anther, each pollen sac contains 1000 mmC, that is megaspore mother cell. What is the total megaspore, not megaspore, that is microspore mother cell, microspore mother cell. If pollen sac contain 1000 mmC, 1 mmC, 1 megaspore mother cell will form 4 spores. If 10,000 microspore mother cell, then what is the pollen grain produced? That will be 4000. So A is the right answer. The next question. If a tetraploid plant contains 48 chromosomes, in its nucleus the number of chromosomes in egg cell and synergid respectively. So as the nucleus it is diploid. So nucleus contains a diploid cell. From this nucleus, one lucky nuclear cell will form a embryo sac, that is megasporangium. Within this megasporangium or the embryo sac, this embryo sac will be n in number. The cells which are present in embryo sac, that is egg cell and synergid, it contains a 24, that is the n number of chromosome. As the nucleus contains 48, then cells which are present in embryo sac will be 24, 24. Hence, the chromosomes in egg cell, that is 24, synergid 24. B is the right answer. The next one, pollen grains are generally spherical, measuring about. So that is pollen grains size, that is 25 to 50 micrometer in diameter. Micrometers in diameter. So A is the right answer. Don't get confused regarding micrometer, millimeter. So it is always micrometer. So next question. Which of the following character is not required for autogamy? Autogamy, it is self-pollination. In order to undergo self-pollination, the flower has to be bisexual, which is not necessary, as both male and female gametophyte will be present in a single flower. Then there is no necessary of pollinating agents. So let's see the options given in this question. Flowers require synchrony in pollen release and stigma maturation. This is true. Anther and stigma should lie close to each other. It is also true. Flowers should be bisexual. It is also true. Here, the D, which is given here, required pollinating agent. As they asked not, in order to undergo autogamy, pollinating agents are not necessary, not required. So, D is the right answer. Then read out the next question. Identify the correct statement. They have given 4 statements, you have to choose correct one. Only one megaspore present towards chalaja remain functional. This is not true. 3 megaspore present towards chalajal and degenerate gradually, that is antipodal cells. Antipodal cells, they will get degenerate. This is true. Each megaspore mother cell directly develops into megaspore. No. 3 will get degenerate, only one megaspore will get formed. Each female gametophyte is 7-celled, 8-nucleated. No, it is 7-celled, 8-nucleated. So this is wrong, this is wrong. Only second option is true. The next one. Next question. Which of the following aquatic plant does not show pollination by water? Here, even though it is present in water, it will not take part in water pollination, but it will take insect pollination, that is water hyacinth. Water hyacinth and water lily. These two, they will take part in insect pollination even though they remain in water. So C is the right answer. The next question, which cell of the female gametophyte is involved in formation of primary endosperm nucleus pen after fertilization? So PEN, that is central cell. Which cell? That is central cell. As the central cell contains two polar nuclei in a embryo sac. Here you will come across three antipodal cell. Here you will come across two synergid cell, one egg cell. The center two polar nuclei which are present in central cell, they will take part in pen formation. Primary endosperm nucleus, that is 3n in number. So D is the right answer. The next question, how many microsporangia are located at the corners of a typical bilobed anthers of angiosperm? How many are there in a particular bilobed anther? That is 4 in number. 4 microsporangia will be seen. That will be situated in corners. So B is the right answer. Read out the next question. Even in absence of pollinators, a assured seed set will be there in, that is cleistogamous flower. As the flower do not open, both gametophyte present in a same flower, self pollination takes place, autogamy takes place. Soon after autogamy, a assured seed set will be observed. So cleistogamous flower is the right answer for this question. Then next question, read out the next question. When pollen grain is shed at three-celled stage, name the cell it contains. Three-celled stage, one vegetative cell and two male gametes. One vegetative cell, two male gamete. A is the right answer. And some of the pollen will shed at two-celled stage, where one vegetative cell and one generative cell will be observed. 17th question. Among the following statement related to pollen, choose the correct one. Statement 1. 40% of angiosperm pollen grains are shed at three-celled stage. It is true. Statement 2. Intine is made up of cellulose and pectin. It is true. And it is discontinuous with germ pore. No. Intine will be continuous, whereas exine is discontinuous, where you will come across a germ pore. So, it is false. Statement 1 is correct. Statement 2 is incorrect. C is the right answer. So, this is regarding your sexual reproduction in flowering plants. We have covered around 23 questions from the first chapter. We'll discuss some more questions in the next video. Thank you.